Welcome, everybody. This is Peter Bauer, Chief Genomic Officer at Centogene, and I really warmly welcome you in these crazy and strange times for our webinar dealing with multiomic markers in clinical diagnostics, where I would like to take you on a tour how genetics and biomarkers, small molecules, can be combined to get to better solutions, better interpretations, and as well better treatment in rare diseases. So we have structured today's webinar in 6 chapters, where we will bring you some information about Centogene first, then highlight how biomarkers contribute to rare diseases and how they can be used in clinical practice. Furthermore, I have an example where biomarkers support variant classification in rare diseases, and I would like to outline how a monitoring biomarker like lyso-Gb1 in Gaucher disease can be used to give rationalities in rare disease treatments. Finally, an outlook into how the field of biomarker discovery and understanding can help us as well to structure rare diseases, understand commonalities, and by that as well find common treatment targets in rare diseases. Now, to start and share with you the vision of Centogene, we are committed to our patients with rare diseases, where especially the diagnostic odysseys need an end with a precise genetic and molecular diagnosis. For that, we have built up a large testing portfolio, and we transform the knowledge into medical solutions by this molecular information, both on the genetic and the metabolic end. These solutions are meant to accelerate development of orphan drugs, because ultimately not the diagnosis is the goal, but the treatment and the help to our patients and their families. Centogene is a global company with headquarters in Boston and Rostock in Germany, and offices in Dubai, Delhi, and Berlin, where we meanwhile work with more than 500 employees in the rare disease business. Our production headquarters is in Rostock, as seen in this small picture on the upper right, where we have established all these state-of-the-art genetic and mass spectrometric technologies to help finding diagnosis and treatment for rare diseases. The company is now 14 years on the way, founded in 2006 by Professor Arne Wolfs. As said, we have more than 400 employees, many nationalities working in our teams, and we have collaborations with many companies working in the orphan drug space. Meanwhile, we have diagnosed more than 400,000 patients from 120 countries, and this information with the diagnosis, of course, leverage back a lot of clinical ad hoc decisions on recurrence risk, on treatment, on prevention, and beyond as well, helped us to develop biomarkers on a commercial level in 10 indications, and dozens of biomarkers in a research environment, where I will give you some insights as well. What is the importance about biomarkers in rare diseases? And in essence, we have first of all to define what a biomarker is, and this is a small molecule usually that is measured as an indicator of a normal or pathological biological process, might respond to pharmacologic interventions, and by that maybe help to measure as well the presence or the progress of disease symptoms, either in total or for subsets of the clinical signs. The biomarkers can be discovered using omic technologies like genomics, proteomics, transcriptomics, and imaging technologies in the genomic medicine paradigm. When do we use biomarkers? Essentially, there's a lot of questions that can be raised with and answered with the help of biomarkers, which is the differential diagnosis of patients, where you want to subclassify according to symptoms. You want to screen and test at-risk cohorts to identify patients with rare disease. You would like to monitor disease progression or measure the response to treatment and thereby guide therapeutic regimen, change treatment if you see that biomarkers inform you that the efficiency has gone down. And of course, you would like to follow up and monitor patients to document in a regular manner that your assumptions and your actions are aligned. In principle, a biomarker can be any small molecules, be it an enzyme activity, so not the molecule itself, but its chemical properties, or the amount or the structure of proteins, could be nucleic acids like DNA and RNA, and of course, and that's something we looked very much in detail into, metabolites that inform us about normal and abnormal metabolic states. Our biomarker pipeline is very generic because we receive samples from patients with known and diagnosed rare diseases where we have established a molecular diagnosis. And we have a large cohort of controls that are either symptom-free or have been excluded for a specific disease and can be used as phenotype controls or validation controls. From these cohorts and patient groups, we have dried blood spot filter cards where we can extract molecules from the blood, whole blood of the patients and the controls. And these samples, small amounts, are used in hybrid mass spectrometry to measure and produce high-resolution data sets with 10,000s of different ions or molecules identified. The bioinformatic of these data sets is focused on identification of abundance or non-abundance of specific molecules that characterize a disease pattern or specific symptoms of patients. And with these markers, then we can go back and validate different phenotypes, similar diseases, sensitivity and specificities of these molecules or profiles. This can be in principle be done within weeks, since samples are usually available in our biobank repository, and the execution of these high-resolution mass spectrometric profiles is a very efficient and fast omic technology. This is depicted here, where already a first application of biomarker screening is shown, where we use the asset of dried blood spot filter cards as a carrier of patient biomaterial to be shipped at very low cost to our laboratory, and within days we can either do enzymatic activity tests or identify validated small molecules, biomarkers in the sample, and whenever the value is abnormal, we can add DNA sequencing to confirm the specificity of the molecule we have measured. The combination ultimately of biochemical and a genetic test gives us a perfect sensitivity and specificity in the screening process and allows to bring down the rate of false positives and false negatives to almost zero. We look into the field of inherited metabolic diseases We know that this is pretty common in all the inherited diseases, so 1 in 1,000 or 1 in 2,000 births is recognized having a metabolic disorder, and these disorders are usually very severe, where 1/3 of the children will not live until the 5th birthday. And so overall, many families and children worldwide are suffering from these metabolic diseases. We look in more detail. We have built up a unique expertise in the most common disorders, which include the sphingolipidosis and other hereditary metabolic diseases, where we can always link a single gene analysis like the GLA gene in Fabry with a specific biomarker, in this case lyso-Gb3, which is elevated in patients with acute and chronic Fabry disease and on normal levels in controls and asymptomatic carriers. We have identified other molecules from the ceramide metabolism or the lysosphingolipid metabolism in Niemann-Pick type AB, in metachromatic leukodystrophy under development, and very prominent in Gaucher disease with lyso-Gb1, where we can see almost perfect correlation of high levels with severely affected patients. Interestingly, there's another gene, fucosyl deficiency, which is the activator of fucosyl ceramidase, that also gives high levels of lyso-Gb1 and can be considered as a differential diagnosis. These diseases are consistently screened in our high-throughput patient programs with biomarker and genetic confirmation, like I have outlined in the slide before. The technology is usually tandem NS because this is very fast and very specific, so the rate of technical false positives or false negatives is reduced to almost zero. And so we have workflows where we can guarantee turnaround times of less than 7 days with this technology established. What is the clinical practice for these biomarkers? And I would like to highlight that we are committed to our patients and giving them support in their diagnostic findings, which is sometimes difficult because exome sequencing might not come up with a very clear variants, but with novel variants where interpretation usually is hampered by lacking knowledge. In these cases, the combination of the genotype and the metabolic phenotype helps us to come to decisions, which I will show you in a minute. For this purpose, we have established our metabolic gene panel, where we always link sequencing of almost 200 genes in metabolic disorders with available biomarker and enzymatic tests. To date, more than 50 of these are established and other biomarkers are under development. We look into that in more detail. You should use this approach when you suspect a metabolic disorder and don't recognize a distinct phenotype that would prompt you to one specific disorder, like morbus Hunter in the mucopolysaccharidosis, or like with very distinct phenotypes, for example, for morbus Tay-Sachs, where you could see the clinical phenotype already pointing to deficiency of hexaminase A. So furthermore, of course, if a diagnosis is very urgent in the critical care environment, or when symptoms are overlapping and you cannot decide on a distinct phenotype, this screening which is complemented by enzymes, especially for single lipidosis and oligosaccharidosis, but as well for steroid lipofuscinosis and mucopolysaccharidosis, is adding value to your genetic screen. Because center metabolic is very fast and you get a precise diagnosis, we have comprehensive coverage on the most important or very large list of metabolic disorders, and as said, the complementary biochemical confirmation in more than 25 diseases, and hence others under development. How does this work, for example, in a family where we have been asked to do Niemann-Pick type AB testing, which is caused by SMPD1 variants, in this case from Sri Lanka? We have identified in the family, and this has been published end of last year, we have identified in this family 2 novel missense variants in SMPD1. While the clinical phenotype was very indicative of Niemann-Pick type A disease, of course, the classification of the variant with no population frequencies, with, of course, no other observations internally, was difficult. However, when we added lysa SM509, the biomarker specifically elevated in Niemann-Pick type AB disease, we could see that the index in the family had high pathologic levels of this biomarker, and this helped us, like shown here, to come from variants of uncertain significance in terms of DNA classification to a conclusive report where we could say these 2 variants clearly impair the function of SMPD1, because as shown in the biomarker level, with more than 5 nanogram per microliters, we have a pathological elevation, which is typical of homozygous or compound heterozygous Niemann-Pick type AB patients. And as you can see as well in the chart, this value is very, very distinct from wild-type controls or heterozygous carriers. Taken together, the combination of genotype, in this case rare variants, and biomarkers is really key for the quality of this diagnostic test. Now let's look into more detail what you can do beyond variant classification with effective biomarkers, and lyso-Gb1 is really a thrilling example how this can inform researchers and clinicians in the best diagnosis and identification of patients. We published 2 years ago, especially the ability of lyso-Gb1 to respond to treatment. And this was a collaboration with our partners in Jerusalem, the Ari Simram group, where we've seen that lyso-Gb1 is presently the most specific and sensitive diagnostic biomarker of Gaucher disease. Moreover, we could even show, and this has been published again with the Simram group, that a reduction of lyso-Gb1 levels is usually seen in enzyme replacement therapy and can help to monitor the efficacy of this very expensive and burdensome treatment for the patients. Nonetheless, it's the most efficient treatment and helps to survive and stay pretty healthy to many Gaucher disease patients. What does it look like? You can see here that we have perfect separation of Gaucher patients shown on the left from patients with other lysosomal storage diseases or Gaucher carriers. And this is as well true for both genders, where we don't have an overlap of normal and abnormal values above 10 nanograms per microliter. But even more important, we could show in these two papers that we can, the level of lyso-Gb1 correlates over time with the efficacy of enzyme replacement therapy. On the left side, in the Arcadia paper, we see a correlation of reduced lyso-Gb1 levels over time in patients under treatment. And on the right side, you can see that the time after initiation of treatment is really followed by significant drops of lyso-Gb1 levels over time, as shown in the right pane or in the individual graphs in the middle. So you can monitor and demonstrate efficacy of drug to guide individualized dosing. And of course, this is very beneficial to patients and payers because you can reduce enzyme amounts and thereby costs if you have achieved steady state. This is also shown here in an example where an individual patient was followed over years with lyso-Gb1 levels, and you can see on the right side that he received in the first years 45 units per kilogram body weight and stayed with lyso-Gb1 levels of below 40 nanograms per microliter. But then after 3 years, the values raised to 60 nanogram per microliter, indicated by the first green arrow, and clinically accompanied by severe bone pain, which led to the higher dosage of enzyme replacement therapy with 60 units per kilogram body weight, and in turn a drop of lyso-Gb1 levels after this dosage escalation. Nonetheless, there was relapse 6 months later, again accompanied by bone pain, and the dosage of enzyme was once again escalated to 75 units per kilogram. Subsequently, the symptoms resolved and the lyso-Gb1 biomarker level dropped again to values that we had during the stable phase shown in the years before around 30 nanograms per microliter. This is very relevant information because it gives rationality to treatment in these patients with rare diseases. And just to illustrate once again, the markers really perfectly differentiating between controls and patients. This is a large validation cohort. They have not seen any control with a level above 10 nanograms per microliter, and vice versa. We have not seen patients with levels below 10 nanograms per microliter. Therefore, in this case, very rarely sensitivity and specificity is just perfect with the data we have gathered. Finally, this is very interesting that you can see it can be as well the other way around. If you are on treatment and then lack the enzyme, which was in collaboration with a partner in South Europe, where due to an export problem, patients treated over years with enzyme replacement did not receive their regular treatment. And after 3 months, in all patients, the lyso-Gb1 level has significantly increased on average 2.5 times within these 3 months, showing that without treatment, the levels quickly go back to very high levels, which show inappropriate or inefficient treatment. So this is a manuscript we have prepared and are ready to submit to share this information as well with the scientific community. Now, so in the end, for us, this is the paradigm where we think lyso-Gb1 should be used clinically. So if you have diagnosed a patient with Gaucher disease, lyso-Gb1 should be monitored at least every 3 months. And then the dosage of recombinant enzyme should be adjusted according to the levels. As you can see, whenever you have an increase, again in lyso-Gb1 levels, you should escalate dosage, while with low levels, you can consider reducing recombinant enzyme and save costs. Now, last chapter is an illustration how biomarkers as well help us to gather new insights into pathophysiology of rare diseases. And this example has been published last year by Claudia Cosma, the head of our biomarker and biochemical research group, and the team where we stepped into a very rare disease, hyaline fibromatosis syndrome, by chance, because actually initially we wanted to characterize morbus Fabr, an enzymatic lysosomal storage disease where deficiency in ASAH1 leads to skin problems where the deposits of the storage products can be seen and cause painful swelling. When we wanted to validate a biomarker in morbus Fabr, which we already knew, which is a ceramide C26, we realized by screening our patient repository that not all individuals with high C26 could be shown to have morbus Fabr mutations. And these ASAH1 mutation negative cases prompted us to do exome sequencing in individuals with high C26 values. And consequently, we have seen that these patients had anthrax 2 mutations, a very rare disorder with the hyaline fibromatosis syndrome, which is clinically quite similar as well, with pulmonary disease and skin abnormalities, and as a joint observation with high levels of C26. What you can see on this slide on top is that these samples of HFS patients on the left in the illustration A have not only C26 significantly dysregulated, so high in HFS and low controls, but there are in the cohort more than 50 molecules with differential regulation that are potential biomarker candidates. And with this capture, we did a principal component analysis, in this case with HFS patients in red, Fabr disease patients in green, and could see that both diseases clustered distinct from our healthy controls. This was very interesting because this suggested that patients with HFS and Fabr disease share some common pathophysiology, because the metabolomic profiles are similar. And you can see as well in the illustration B that the arborization of the individuals really clustered all HFS and Fabr disease patients in one part of the tree, while all controls were pretty much aligned on top, which tells us that these two diseases are very similar, or at least have commonalities in their metabolic profiles, which is quite surprising because, as said, Farber's disease is an enzymatic defect, formerly a lysosomal storage disease, while HFS is a glucogen receptor deficiency where the shared pathophysiology is not obvious. And intriguingly, out of 5,000 compounds that have been identified in all patients of both cohorts, 81 are shared like C26. So we have now the potential to describe rare disease by giving an overlap and a differentiator for two distinct genotypes with overlapping phenotypes. You can easily understand that this type of research, and later as well target identification, helps us to delineate shared therapeutic targets as well for diseases that are not delineated jointly yet. And this has, besides the diagnostic opportunity, as well a huge impact into drug testing, drug development, and the potential to repurpose orphan drugs in other indications. So to summarize today's webinar, I think I could share with you good evidence that high-resolution metabolomics is a natural partner for interpreting exome and genome sequencing data in rare diseases, especially when dried blood spot filter cuts are used, because this biomaterial is the source for both omic technologies in our hands. Biomarkers, especially metabolomics, can be used for generic support in variant classification. I've shown you the example of Niemann-Pick type AB, where variants that usually are classified uncertain clinical significance can be interpreted as likely pathogenic or pathogenic based on this outstanding stepping stone evidence from metabolic disturbances. These markers have the potential to elucidate state and trait biomarkers in rare diseases and thereby help us to develop understanding of disease courses, severity, and response to treatment. And of course, like in the last example, I've shared with you our understanding and insights into pathophysiology and potential treatment targets in rare diseases, which can be delineated from high-resolution metabolomics in genetically defined cohorts. Taken together, I think that's, for me, kind of the leverage for genomic medicine of the future, where we can inform both sides from high-resolution omic technologies and thereby come to more precise interpretation and prediction in patients with rare diseases. At this stage, I thank you for your interest and your attention, and I'm happy to take questions now. Would invite you to use the Q&A form of this webinar software and just drop any questions which I will read and answer in the next minutes. On top, be aware that we will send you a questionnaire after this webinar where we ask you for your feedback in terms of expectations met, format, content, and anything you would like to recommend or suggest for upcoming webinars to give us guidance about your interest. Thank you for that, and having said that, I jump now into the Q&A. First question is, how do we select which molecules can be considered as potential biomarkers? It's a very good question, because what is the characteristics of a good biomarker? It should be a very stable molecule. So we want to see biomarkers stable over time in filter cards so that we can measure them as well in bioarchive. We want to have molecules that are very specific usually, so for distinct diseases, and biomarkers that have the potential to be as well developed in standard ELISA tests, of course, help us to go into large screening programs. On the other hand, of course, as well, a biomarker that might be more tricky, not able for ELISA screening, can be very useful if the biomarker is close to the real pathophysiology of a disease and not only just a derived marker, thereby helping us to understand and screen for potential therapeutic drugs. And so depending on the intended use, the characteristics of biomarkers are rated differently. For a clinical biomarker, it should be very robust. The specificity should be, and sensitivity, of course, should be very high. So it should help to diagnose patients and not come up with many false positives. This was as well a follow-up question. So what is the sensitivity and specificity of these tests? Well, there's not a general answer to all biomarkers, but usually the sensitivity is above 99.9%. If we don't reach that and would expect to lose more than 1,000 patients, we would add maybe a second marker for screening so that we can include the missing patients as well, and then do the confirmation, as I told you, by sequencing, which gives a very high specificity. So the combination of ultra-high sensitivity with the biomarker molecule and the confirmation by genetics is a perfect tool to avoid false negatives and false positives. Another question is whether microRNAs can be considered as biomarkers for rare diseases, for example, as well in Gaucher disease. Yes, of course. So all molecules that are highly correlated with a phenotype have the potential to be considered as biomarkers. Our experience is that the robustness of how to measure these markers and how to solve logistics for this analysis is crucial to make use of biomarkers. And there, the filter card is very good because we have a stable material that can be shipped at low cost from all countries of the world virtually. And so it's more a question of pre-analytics and less about the molecule. We have looked into RNA molecules on filter cards. They are stable within the first days, but of course, the quality after 2 weeks is impaired, and therefore we prefer to have small molecules that are more stable. There's a question whether we have different blood level of biomarkers for different ages, probably ages of patients. It's not really age-related, what we've seen, at least for the markers I've presented to you, but very often it's severity-related. So the biomarker should reflect the activity of a disease, and that's seen in many of these markers I shared with you, which means if there's no treatment and disease severity is piling up over the years, of course, we see as well a constant increase in biomarker levels in different ages, but this is rather reflected by the lack of treatment and not by the age of the patient. So there's a question on the enzyme assays in the central metabolic test, whether this is directly linked to the genes or the variants we see in sequencing. Of course, we don't measure all the enzyme levels in all panel requests, because if we don't see a genetic variant, there's no need to do enzymatic analysis. However, if we have variants, we can start looking into the specific enzyme activities and validate the genotypes we've seen. Another question is when to do the biomarkers. Can they be done in the newborn screening for selected metabolic disorders? This is as well a very good question, and we have looked in a couple of the more prominent biomarkers. At this purpose, we see that, for example, biomarkers for Gaucher disease and for Niemann-Pick disease show up very early in life, and we have already elevated levels at birth, usually. Whether this is a necessary screening test in the neonatal, I cannot yet tell you, because we don't have many data on that, but I know that a couple of neonatal screening studies consider to include lyso-Gb1 in Gaucher disease, and consider as well to include lyso-SM509 for Niemann-Pick type C disease. The question is always, how treatable are those diseases, which is better to be answered for Gaucher disease than for Niemann-Pick type C disease. But in principle, the abnormal levels start early in life. There's a question whether this session can be found in our video section as well. This is possible, so we will post the recorded webinar within the next days. And just check. I think essentially that's the questions I received. Maybe the last one is there differences in different ethnicities? So do we see that biomarker levels might be higher or lower in subpopulations? That's as well a very relevant question. We have done such an analysis in Niemann-Pick type C and in Gaucher disease, where we didn't see ethnicity differences. Rather, we've seen that the biomarker levels in Gaucher disease correlate with the pathogenic variants, so from milder to more severe variants with lower or higher biomarker levels, and usually it's correlates with the clinical pictures, but not with ethnicities. Good. Now, I think this is what has been posted on our Q&A. Once again, I thank you for your attention, and I hope that this helped you to prepare for your next difficult cases. Contact us if you see these patients and think biomarker analysis or metabolomic screening might help to come to a better understanding. And for today, I wish you all a very good day. Stay healthy, and happy to connect with you soon again in the next webinar, or whenever possible as well in person. So thanks for your support, and have a good day. Bye-bye.